Good? All right, you guys, your attention, please. We're going to talk about chapter 9. We already talked about DNA replication. We already talked about gene expression. Please pay attention here. There's a lot of information coming your way. We're about to talk about operons, and they're involved. You'll see what I'm talking about once we go through them. So I definitely need your attention here. The Gen Bio Review PowerPoint, and at the end of it, you guys have some sample questions that I want you to make sure you work on. Make sure you do the General Biology Review PowerPoint and the PowerPoint questions. You already have answers for them. So I gave you answers for them on Canvas. So make sure you know how to do the DNA replications, the enzymes involved, and so forth. To quickly recap what we talked about. We talked about on, when was that? Last Thursday, right? Mm-hmm. We talked about gene expression. We also talked about DNA replication. So we said that DNA replication is basically the copying of the DNA to DNA. There are a whole bunch of enzymes involved. You need to know what the name of the enzymes are, when do they come in the process, and what do they do. For example, topoisomerase opens the origin of replication. Another topoisomerase untangles the DNA ahead of the fork. Then the next enzyme is going to be helicase that unzips the DNA by breaking down the hydrogen bonds. The next enzyme is the single, no, it's actually not an enzyme, it's actually a protein. The single-stranded binding protein keeps the DNA single-stranded. The next enzyme is primase. It makes the primer. Remember, the primer is RNA. And that's for DNA polymerase 3. DNA polymerase 3 will come in and make DNA. So it's in the name, DNA polymerase 3. And then DNA polymerase 1 will come in and say, we don't need this primer anymore. So it will remove the RNA primer and replace it with what? With DNA. Very good. Then the final enzyme is DNA ligase, and it seals the gaps. If you know that information, you are good to go with that one. You also need to know the difference between leading strand and lagging strand. You need to know the difference between continuous and discontinuous. What are Okazaki fragments? What is semi-conservative replication? DNA replication is also bidirectional. That means it goes in both directions for efficiency. Those are the things you need to know for DNA replication. Then we followed up with gene expression. We quickly talked about the difference between gene expression in prokaryotes versus gene expression in eukaryotes. And the main difference has to do with where they happen. Gene expression in prokaryotic cells happens in the cytoplasm. That's it. Both transcription and translation happens in the cytoplasm of bacteria. Gene expression in eukaryotic cells, like your cells, we call that compartmentalized. Say that 10 times, right? Compartmentalized means, it's a fancy word for meaning it happens in 2 locations. Transcription and RNA processing happens in the nucleus of eukaryotic cells. But translation happens in the cytoplasm. So in eukaryotic cells, you have to finish transcription all the way before you start translation. Why? Because they happen in 2 different areas. So it's like transcription happens in this room, translation happens in the other room. That means I have to finish transcription completely before I start translation in the other room. You don't have to do that in bacteria though. What I'm about to tell you is some of the differences between bacteria and eukaryotic cells. Look at this. Bacteria has what is called co-transcriptional translation, meaning transcription and translation happen at the same time. They can do that because of both of them happening in the cytoplasm at the same time. See, you gotta catch this. Because prokaryotic gene expression is not compartmentalized, meaning because it happens in one location, you can translate the message at the same time you're making the message. So if this is the mRNA, look at this here please, look up here please. As the RNA polymerase is making the RNA, the ribosome is already binding to make the protein. You can do that in bacteria. You can't do that in eukaryotic cells. Because in eukaryotic cells, transcription happens in the nucleus, but translation happens in the cytoplasm. So in eukaryotic cells, you have to have the message already done before it leaves the nucleus, goes to the cytoplasm for translation. Prokaryotic cells do not have to do that. They both happen in the cytoplasm. So as the RNA polymerase, the enzyme that's making RNA in transcription, is making the RNA, the ribosome is already binding and making the protein. It's amazing how that works, and it's very fast, very efficient. So I gave you basically the name of the enzyme involved in transcription. Thank God, there's only one. In DNA replication, we made a whole list, right, of enzymes. In transcription, there's only one enzyme, and it's RNA polymerase. That's it. RNA polymerase will sit on the gene and transcribe it into RNA. Now, every gene has a region called the promoter. Listen to this one, please, because it will introduce what we're about to talk about in the next slide. Every gene has a region called the promoter. We can think of the promoter as the light switch of the gene. It either turns on or off gene expression. And it will always be positioned right ahead of the gene. So you have the promoter element and then the gene. RNA polymerase will bind to the promoter to activate gene expression. Let me give you an analogy. The light switch by the door is the promoter. When I walk into the room, I will hit the light switch to turn the light on. The light switch is the promoter. I am RNA polymerase. And when I touch the light switch, gene expression, the light, turns on. Let me ask you something. The first time you walked into this room, you've never been in this room before, hopefully, right? Where did you think the light switch was?
By the door.
Back there, hiding behind the counter over there, or by the door?
By the door.
It's always going to be by the door. The people that build these rooms or your home or whatever, they put the light switch by the door so you don't have to go into the dark room and stumble and fall and break your neck looking for the light switch. Now, they can double up on the light switch, that's a different story. But all light switches are by the door. All promoters are at the beginning of the gene. The light switch of the gene is right at the beginning of the gene. You don't have to go looking for it. We know where it's at. And who binds to the promoter again? RNA what? Polymerase. And when it does, it activates transcription or gene expression, the making of RNA. Now, the promoter is it's in common. How that promoter works is in common with eukaryotic cells and prokaryotic. The prokaryotic cells have promoters too. But there are some differences. Hang in there, we'll talk about it. And that's where the fun begins, the differences. All right, AUG is the start codon. AUG is the first codon that tells the ribosome, hey, start translation here. It is called the start codon. Interestingly, in eukaryotic cells, the first amino acids that's brought by the start codon is always going to be methionine, met. But in prokaryotic cells, bacteria, it's F-met, formal methionine. So in eukaryotic cells, it's methionine. In prokaryotic cells, it is referred to as F-MAT, methionine. So if I give you a protein sequence and it starts with F-met, and I ask you, which organism produced this protein, what would you say?
Prokaryotic.
It's bacteria, prokaryotic, or archaea. So that's how you distinguish between them. If it starts with methionine, it's eukaryotic. If it starts with F-met, It's prokaryotic. That's another difference. All right, here's another difference. Prokaryotic cells do not have introns, so there's no need for splicing. We talked about splicing when we talked about gene expression, but we were talking about eukaryotic cells when we were talking about that. Eukaryotic genes have introns that must be removed. The removal of introns is called splicing. You don't need to do that in bacteria because they don't have introns. That's another difference. Here's another difference. For each mRNA in eukaryotic cells produces one protein. That's not the same in prokaryotic cells. You're about to see it. In prokaryotic cells, a single mRNA can contain the information of several genes in a row. So, in eukaryotic cells, you have an mRNA for catalase, for example. You have an mRNA for a membrane protein, for example. But in bacteria, a single mRNA can be used to make multiple proteins because the genes are arranged differently. The genes are arranged in a row. So bacteria is interesting. Bacteria is like a neat person, a person who's very clean and neat at home. I can't be messy, I can't be messy, I can't be messy with my genes because I don't have space. The bacterial chromosome compared to your chromosomes is relatively small. You have 3 billion bases of DNA. You can afford to be messy with your genes. Meaning, if there are 3 genes involved in metabolizing, for example, glucose, one of the genes could be on chromosome 1, the other gene could be on chromosome 10, the third one could be on chromosome 20. In eukaryotic cells, they can be on different chromosomes. It's okay. We have the real estate for that. We have 3 billion bases. We can afford to be a little messy with our genes. Not bacteria. Bacteria says, I have 3 genes that I need to use to metabolize lactose. We're about to talk about that. And this is where your attention is needed. All right? I have 3 genes that will metabolize lactose. I'm going to keep them right next to each other. I can't afford to be messy. So, prokaryotic cells arrange their genes called structural loci or locus. For example, the 3 genes involved in metabolizing lactose in bacteria, they will be right next to each other. They're called tandem. They're right next to each other. On top of that, you're going to also have the control elements right next door to them too. The control elements are the promoter and the operator. We'll talk about it. And then you're going to have the regulator gene nearby. It doesn't have to be right next to them, but it's nearby. The whole thing together is called an operon. So what we say is this: prokaryotic cells arrange their genes in clusters called operons. We only see them in prokaryotes. We don't have operons. They have the control elements, the regulatory elements, and the genes as a complete cassette or cluster right next to each other. So it would look like this. Look up here now, please. You have the promoter. That is where RNA polymerase binds to turn on gene expression, transcription. You have an operator segment. We'll talk about it. You have the regulator. And you have what's called the structural genes. So look, bacteria likes lactose. I don't think any bacteria will say no to lactose there. A lot of them do actually. But they don't always eat lactose. Simply, what if I don't give it lactose? What? It's not gonna just die. It'll just eat something else. So you like Chick-fil-A. It doesn't mean you always ate Chick-fil-A. You can eat something else. But if I give you Chick-fil-A, you'll go for that. So let's talk about a bacterium that likes lactose. But let's put it in a situation where I'm not giving it lactose. So it doesn't need to metabolize the lactose because it doesn't have lactose. But it will survive off of other food until you give it lactose. So look what's happening here. You have 3 genes. All 3 of them are involved in metabolizing the lactose. All 3 of them. I need your attention, please. All 3 of them are involved in metabolizing lactose. And they're all positioned right next to each other. But look what's happening with these 3 genes. Right now, their expression is in the off position. Because it doesn't have lactose. So the first scenario is, I did not give the bacteria lactose. So it does not have to make the enzymes that metabolize the lactose. Why? Because it doesn't have lactose. You can't eat Chick-fil-A if I don't give you Chick-fil-A. You see what I mean? Mm-hmm. So you have to wait until I give you Chick-fil-A before you eat Chick-fil-A. Not that I have any money in Chick-fil-A there, right? But that's the whole idea. Lactose, I'm not going to be able to eat the lactose until he gives me lactose. So I'm not going to turn on the genes involved in lactose metabolism until I get the lactose. So in this position right now, this operon is said to be off. This is equivalent to you walking into this room with the light off. The only option is to do what? Turn the light on. That's why this operon is called inducible. Listen to it carefully now. If you're listening to me, you're gonna thank me later that I warned you to listen to me. Don't rely on this video. I may decide not to post it tonight. So the inducible operon is in the off position first. And it's called inducible because I can turn it on. Are we catching that? Mm-hmm. So how does it stay in the off position? Here we go. The regulator gene will make what's called a repressor protein. The regulator gene will make a protein called the repressor. This protein, once it's made by the regulator gene of an inducible operon, it is already in the active form. It's made in the active form. Active form meaning what? Meaning this. Its shape is perfect for it to run and bind to the operator. So the shape of the repressor protein is in the active form, meaning it's perfect, and it will run and bind to the operator segment of the operon. When it does, it will block the binding of RNA polymerase to the promoter. This is like me going to the light box, the light switch. And before I go to the light switch to hit the light switch on, Leo goes to the light switch and blocks it. You see? Leo puts his hand on the light switch. That means I can't turn it on. So he's the repressor. The only way for me to turn the light on is to do what with Leo's hand?
Move it.
Remove it. You see that? So watch this. When the repressor is sitting on the operator, it will prevent RNA polymerase from sitting on the promoter and activating gene expression. That means this gene, all these genes will be in the what position? Off. It'll be in the off position. And they will stay like that. This is an inducible operon. Its normal position is off. So, it's like Leo always having his hand on the light switch. I can't turn it on until someone comes and removes his hand. So meaning, until someone comes and removes this repressor protein, will RNA polymerase bind to the promoter and activate gene expression. Who's that someone? Oh, look at this. Lactose itself. I mean, this is amazing. You think they're just bacteria. What do they know? They know a lot. Look what they do. I will only turn this operon on when you give me lactose. Why do I go make the enzymes that digest lactose and then find out that I don't have lactose? I just wasted resources. I wasted amino acids, ATP, and a lot of effort. So look what they do. They keep the operon off until you add lactose to the media, because lactose itself is the one that's going to knock off Leo's hand off that switch. Lactose is the inducer of its own operon. I mean, that is clever. That's how your bacteria is going to know that you gave it lactose. Lactose will bind to the repressor that's sitting on the operator, and it will change its shape. And the repressor will fall off. Now RNA polymerase will say, here we go. I got access to the promoter now. The repressor is no longer there. RNA polymerase will sit on the promoter, activate gene expression. Now, we have to understand what these enzymes do. They all are involved in the metabolism of lactose. One of them lets lactose into the cell. It's called lactose permease. It allows lactose into the cell. That's one. The other one modifies the lactose to make it a little bit more delicious. And then the third one breaks it in half, because lactose is a disaccharide sugar. They're all involved in lactose metabolism one way or the other. So you only activate them when lactose is there. Why do I need to go and produce these enzymes and find out there's nothing for me to metabolize? I just wasted energy and amino acids and all that. So give the control to lactose. Only when you add lactose will this operon turn on. That is clever. So lactose will bind to the repressor, knock it off. The RNA polymerase will bind to the promoter, activate gene expression of the structural genes, and all of these enzymes are designed or used in the metabolism of lactose. So look what's going to happen now. We're about to turn it back off. These enzymes say, yummy, yummy, delicious lactose. More yummy, yummy lactose. More. And they say this, I wish I had one more. You know, it's like eating candy, right? You eat, then you say, oh, I wish I had one more. Maybe it's just me, I don't know. And then you do find one more. Look at that. Once it eats this one, the repressor reforms and goes back and turns off the operon. Why? Because it says this, if you have to resort to eating this one, that means you ran out of lactose. That's what it means. Why do I need to keep the operon on to metabolize lactose that you just ran out of? So once it consumes this lactose here, the repressor reactivates. It sits on the operator, turns it back off. Can I turn it back on? Yeah. When you add what again?
More lactose.
More lactose. Is that clever or what?
Yeah.
When I first learned this, I almost literally, maybe call me a geek or whatever, it's okay. I almost literally fell off the chair. That's amazing how that works. I hope you feel the same enthusiasm. Because if you don't, the next slide is going to blow your mind away. Do we understand this one before I switch it up on you? I need questions if you have them. I'm going to flip it on you. So I need you to know this one before I flip it. Questions, please. So on Thursday after the exam, if I call one of you to present this to the rest of the class, and I've been known to do that, would you be able to do it?
Hopefully.
Good, hopefully. So make sure you have it down by Thursday, please. Okay, let's do the other one. The other one is called the Repressible Operon. The Repressible Operon is equivalent to you walking into a room where the light is already on. So what's the only option now? The only option is to turn it off. Now why? Why would you use an operon that is always making something? See, the operon again will have structural genes that will produce enzymes, but in this case, it's making something. See, in the previous case, the inducible operon, that was catabolic, it was breaking things down. But this one is making things, anabolic. It's because it's making an important molecule. Arginine is one of the 20 amino acids. No bacteria can say, no, it's okay, I don't need arginine. The bacteria that says, I don't need arginine, is a dead bacteria. It needs arginine. It needs histidine. It needs the 20 amino acids like you do. You can't say no to arginine either. So look what bacteria does. It's, again, fascinating. I must have arginine. No doubts about it. I must have arginine because arginine is one of the 20 amino acids. I cannot make proteins without arginine. So, I'm going to put it under the control of a repressible operon. A repressible operon is always in the on position because of what it's making. It's making an important molecule. I tell you, this is equivalent to you having access to water all the time. You better. You may not have water in your backpack, but you're probably like 10, 50 feet away from water. Maybe it's a fountain or whatever. You're not that far from water. There's water right here in the sinks right there. You always have to have access to water because it's precious. Without water, you don't survive. No different from arginine. Without arginine, I can't make proteins and I cannot survive. So, I'm going to have it under the control. I hope you're listening to me. I'm going to have it because usually what I see the people who are not listening, and those are the ones that are presenting on Thursday. Those are the ones that are presenting on Thursday. So I gave my secret away. So the arginine is an important metabolite. I must have it. So we're going to put it under a repressible operon. And we're going to do this. The bacteria is going to do this. Make arginine, make arginine, make arginine, make a whole mountain of arginine. And only when I have enough arginine will I turn this off. This is completely different than the inducible one. The inducible one is this. I will only turn on when you give me lactose. But this is different. I will only turn off when I have enough arginine. So this is equivalent to you going to buy water. Buy water, buy water, buy water. Do we have enough? Yes. Okay, then stop buying water. And then when you consume the water, eventually when you reach for that last bottle of water, you're going to realize, oh, I better go get some more. Well, it's the same thing. Make arginine, make arginine, make arginine. Do I have enough? Yes. Okay, turn off the operon. How do you turn it off? Watch this now. The repressor protein that's made by the regulator of a repressible operon is made inactive. That means it does not bind to the operator right away. You don't want it to, because if it binds to the operator, you're going to turn it off. You want it on. So you want the repressor protein not to be active. You make it, but it will not bind to the operator, not yet, because you want it on. Now watch this. Once you have enough arginine, look what's going to happen. One of the arginines is going to go bind to the repressor and activate it. That's when it binds to the operator and turns it off. Because by then it says, oh, the only time I will have the arginine bind to the repressor to activate it is when I have enough arginine. So once it makes enough arginine, one of the arginines will bind to the inactive repressor to activate it, to sit on the operator, to turn the operon off. Because by then, the bacteria has enough arginine. So we call the arginine the co-repressor. We call lactose the inducer. But in this case, we call arginine the co-repressor. It works with the repressor to turn off the repressible operon. Now watch. The cell will say, I need this arginine to make this protein. I need this arginine to make another protein, and another protein, and another protein. And again it goes, I wish I had one more. Well, here's one more. When it goes to reach for this one, the repressor reforms, inactivates, because by then it realizes, oh, we ran out of arginine. Because he will not reach for this one unless he ran out of these. So the repressor will fall off, reactivate the operon to make more arginine. I mean, that's fascinating. The control is amazing. What is more amazing is which ones are under inducible control and which ones are under repressible control. If you're breaking something down, it's going to be inducible, because you'll only initiate the induce the operon when you add that metabolite. I don't need to add lactose. I don't need to eat lactose. So I can live off of something else until you give me lactose. But once you give me lactose, I'll activate the operon so I can eat it. So inducible operons are usually for catabolic reactions, breakdown reactions. Repressible operons are typically for, what do you think? What reactions is this? You're making something, so what is that called? Anabolic, yeah. And it makes sense, because the arginine is crucial for the bacteria to survive. It will have it under repressible control. Only turn off when you have enough arginine. Makes sense. Questions on that before I change gears? Questions on it?